Hi everyone. Today we are going to talk about the adaptation of Chlamydamonas nivalis to growth in low temperature environments for carbon dioxide fixation on Mars. This presentation will be given by Xicheng He, Kaichao Yang, and Gloria Zhou for our EECE 506 Bioprocess Engineering class at Washington University in St. Louis. Starting off with some background on our topic, what's so special for Mars? As space travel, research, and planetary missions continue to rise in popularity, with billionaires like Elon Musk investing large sums of money to send people to the red planet, questions of how to acquire sufficient supplies of food and fuel have become increasingly important. Among the resources available on Mars, its high atmospheric concentration of CO2 provides a promising foundation for the in situ production of food or fuels using organisms that can fix this carbon source. For the application of biotechnology for carbon fixation on Mars, the key challenge that needs consideration is the harsh environment, which is totally different from Earth. Mars receives much less sunlight, and the average temperature on Mars is extremely low, around negative 63 degrees C, and it has a more viscous fluid environment. These harsh environmental conditions will cause harm to the common bacteria found on Earth by slowing the rate of biochemical reactions, reducing enzyme activity, decreasing membrane fluidity, and denaturing proteins. Several types of microorganisms are attractive and may have potential to be applied. Macroalgal species can complete CO2 conversion, yet cannot survive in low temperatures. Saprophytes, on the contrary, can function at low temperatures, but most are unable to fix CO2. With all these factors considered, Chlamydamonas nivalis is an ideal candidate for this project. C. nivalis is a red-colored photosynthetic green algae that can maintain active photosynthesis at 4 degrees C. This species may also be able to confer resistance to UV radiation and achieve carbon fixation, making it attractive to be used in CO2 conversion on Mars. However, challenges still exist when directly applying this species on Mars, since the temperature on Mars is extremely low. Aeroprone PCR could be a biotechnology to further improve C. nivalis. This method introduces random mutations into a defined segment of DNA by promoting the misincorporation of nucleotides, destabilizing non-complementary base pairs, and reducing the specificity of the polymerase. By applying this method, we hope to find the variant of C. nivalis that could grow and fix CO2 at extremely low temperatures.
Next, we'll describe some of the methods and materials for our particular project. So starting off, for the design of our project, 3 project aims were developed. First, we want to use error-prone PCR to select for a variant of C. nivalis that can grow at temperatures of -15 degrees Celsius, which would bring us one step closer to the goal of having a viable production strain for the low temperature conditions of Mars. Next, we aim to develop a predictive model for estimating the lower temperature limit of viable growth for our strain. Since our experimental conditions only specify decreasing the temperature to -15 degrees Celsius, but temperatures on Mars can get as low as -63 degrees Celsius, we want to use computer simulations to estimate just how low we can reduce the temperature to before growth is completely shut down within the cells. Lastly, we want to further use error-prone PCR to select for strains that can grow under increasing concentrations of CO2, ideally up to 95%, which is the atmospheric concentration of CO2 on Mars. Since C. nivalis is a strain native to planet Earth, where the atmospheric concentration of CO2 is about 0.04%, some degree of adaptation would be necessary to successfully transition to growth and production on Mars. As with any other living organism, these microalgae will need their own food in order to grow. Thus, we intend to use an adapted form of the bold 1NV medium recipe from the culture collection of algae at UT Austin to incorporate additional salts and glycerol to lower the freezing point of the medium so that cells can be grown at lower temperatures. For error-prone PCR to be carried out, we must identify a region of the genetic sequence that we want to specifically target. Here, we have listed various genes that were previously identified to be either up or down-regulated in C. nivalis as compared to a standard high-producing microalgae, Chlamydamonas reinhardti. One particular gene of interest is labeled contig 263.3, which was identified as a desiccation-related protein that is down-regulated in C. nivalis. Since desiccation involves the removal of moisture, the downregulation of this gene likely plays an important role in helping the cells to retain their moisture and survive under low temperatures. For the specific PCR design, we plan to use the recipe from Chen and Arnold, published in 2013, which includes ingredients such as manganese chloride and unequal ratios of DATP to DGTP, DCTP, and DTTP, which can disrupt the process of gene amplification to introduce mutations, hence the name error-prone PCR. The thermocycler settings used to carry out these experiments are shown here on the right and involve a series of steps similar to standard PCR, where the mixture is first heated to a high temperature to denature the double-stranded DNA into two single strands, to which the primers can then anneal to the sequences and begin creating the complementary strands during an extension period. This denaturation, annealing, and extension process is repeated for 45 cycles, longer than typical reactions, which are carried out for about 30 cycles, to produce the amplified gene sequences of interest. Specific conditions for growth are included here. We want to begin our experiments by growing cells in our modified medium at 22 degrees Celsius. During mid-log phase, which is when cells are most robust and growing exponentially, we will then subject them to cold treatment to identify strains that exhibit the best growth, starting with 4 degrees C and gradually decreasing with each round of PCR and growth to eventually reach minus 15 degrees C. Growth will be measured by looking at OD measurements for a wavelength of 480 nanometers. And lastly, in reference to our third aim for improved growth in higher CO2 concentrations, we will do a similar set of experiments, but with increasing CO2 and a fixed temperature of minus 15 degrees Celsius. If this project is successfully conducted, we would expect to see a significant growth of C. nivalis at minus 15 degrees C, as measured by greater difference in the final versus initial OD. We would also expect to see greater product concentration with increased CO2 up to a certain threshold, the specific value of which is to be determined during the actual experiments.
Another important aspect of evaluating our project's feasibility is to consider the economics of design. So in this next section, we will discuss the key costs and the sources of revenue to estimate two important financial parameters that can help in deciding whether to pursue this project. We calculated the cumulative net present value and the return on investment to show the feasibility of this project. The calculation assumed that the lifetime of the equipment is 10 years, and each year we can produce 20,000 tons of biomass with the lipid yield of 23%. Additional assumptions are used in our calculations are listed here. Two specific economic parameters that are useful for determining whether the project should be a go or no go include the cumulative NPV, which provides a measure of how much the project is worth in present-day dollars, and the return on investment, which evaluates how much profit we could make for the amount of money invested into the project. Based on our tabulated calculations, the NPV was determined to be about $335,000, and the ROI was about 1.2%. These estimations indicate that
our project has the potential to be profitable for our set of assumptions. Here's some future work that could be made to improve the feasibility of our project. First, in our project, the designed culture temperature of nivalis is minus 15 degrees C, and we want to further lower it to minus 63 degrees C, which is the ambient temperature on Mars. Other factors such as low pressure and dim light on Mars will also be considered. Lastly, we can consider how materials transportation and the dewatering steps for product extraction would affect the cost to arrive at more holistic evaluation of nivalis product generation on Mars.